Okay, so in chapter 10, now if you're confused, we don't do chapter 9 in our books. We just skip over that, so don't worry if you've missed something. But in chapter 10, we're going to talk about this really interesting thing called cell reproduction. And why I think of it so interesting is because here at a very basic level, we start to see one of those extremely important characteristics of life, that ability to make copies of themselves. And I think I said early on in the semester, I always wondered if maybe there was some life form that evolved on Earth, but for some reason the reproductive part, that characteristic didn't evolve, and these organisms would have lived and then died, and that would have been it. We never would have heard about them presumably again. That would have been a pretty important thing to keep in those characteristics of life, that reproduction, that perpetuation of the species, right? And so that's what we're going to talk about in chapter 10. In chapter 10, we're going to talk about reproduction in a very simple way. So what I mean by that is the kind of reproduction that goes on in chapter 10, which we call mitosis, is mitosis is cell. I'm typing it. Mitosis is cell division as if you were making a copy on a copy machine. So in other words, the cells that arise out of mitosis, you can see we got pictures of cells here in different stages of mitosis. It's really kind of cool what they can do with microscopes now. They can get individual cell components to fluoresce in different colors, like here over on the left. There we go. Here over on the left, the nucleus, the chromosomes have been colored, and then the microtubules and the spindle fibers and all that. It's really kind of neat. But what we end up with the end of cell reproduction is an exact copy. So we're creating clones of one another. Now in chapter 11, that's going to be very different. In chapter 11, we're going to talk about a cell reproduction where we actually end up creating different types of cells, genetically different types of cells. Chapter 11 is going to lead us to understand how gametes are made, sperm and egg, and how we get all that variability. I always tell my Bio 2 students when we look at mitosis and meiosis in the different life cycle of the organisms we examine, to think about meiosis as generating cells that are variable, right? And think about mitosis, which we'll talk about today, as generating lots of cells. So one is more quantity and one is more variability. So we're going to deal with the quantity one today. Now, as you can probably imagine, there's a number of reasons why cell reproduction is important. One is the one I mentioned before, for the perpetuation of the species. So as you see in the upper left, it's important for reproduction. Cells are involved in that. More so in the next chapter, we're going to talk about those types of cells. But once that occurs, then of course what we do is we are multicellular organisms. So through growth and development, we had two cells, and you can see in the picture in the upper right, another really cool electron microscope picture. These are two animal cells getting ready to divide. You can see what happens, and we'll talk about this a little bit later. It's kind of cool, though, especially when you look at this picture. The way animal cells finally pinch in half, it's like drawing a purse string or drawing the string around my little crown bag that I bring with all my pens and stuff in. So this right here, what you're seeing is like a cinching of proteins. They basically keep tightening and keep tightening and keep tightening until it finally pinches the cell in half. This thing on the right-hand side is really kind of cool. It's got an interesting name. It's called a morula. And when organisms develop, right, when Junior develops, first, of course, it's one cell when the sperm and the egg, and then those cells divide like you see in the picture, and then they divide a few more times. And very early on in development, what we get is kind of a mass of cells. Morula comes from the Latin word for grapes. So groups of grapes. So you can see the reason why it kind of has that name of a morula. Now that morula is going to develop well beyond that, but that's something we talk about in Bio 2. All right, now I said we were going to talk about cells in terms of reproduction and the way we do it in chapter 11, but I do need to talk about a form of reproduction that occurs this way that we're going to talk about in chapter 10 in a very select group of organisms, and those are the prokaryotes. All right, the prokaryotes don't engage in what we're going to talk about in chapter 11. Instead of me keep saying chapter 11, let me say the word. Chapter 11 involves a type of cell division known as meiosis. Remember, meiosis makes variable cells. Now, prokaryotic cells don't do that. Meiosis involves a lot of genetic shuffling, genetic variability. That's why organisms that are non-prokaryotic, there's so much variability in those. And there's a lot of variability in prokaryotic cells too. But the way they undergo organismal reproduction, which is, by the way, if you think about it, simultaneous with cellular, because prokaryotes are single-celled organisms, is a little bit different. Relatively simple, though. So let's follow up at the very top. We have an E. coli cell. We hear about E. coli all the time, usually in the news for bad reasons, the pathogenic strains of E. coli that get into food and cause food poisoning, all that sort of stuff. But we're going to make a copy of that E. coli. The little purple thing that's all round up inside is a chromosome. If you remember from our discussions, chapter 4, I think it was, we talked about some of the difference between prokaryotes and eukaryotes, and we said one of them was that eukaryotes had multiple chromosomes. We have 46 or 23 pairs, whereas prokaryotes have one singular chromosome. That is one single chromosome. If you pulled that whole chromosome out of that cell and unwound it, it would be one chromosome in a singular loop, one piece of DNA in a singular loop. So in order to make copies of cells, and this is something that's going to be carried through when we talk about eukaryotic cells, and it actually makes kind of perfect sense, before the cell divides, before the cell physically becomes two entities, and every cell needs genetic information, you're going to have to divide the genetic information first. And that's what you see there where it says origin of replication, the part in the green. What happens in this case is we actually start making copies of the DNA and moving in both directions. Mother Nature is very efficient. Rather than making a copy going one way, that origin of replication spreads out from one spot in both ways around the loop until they meet on the other side. Now, it's a little hard than seeing meeting on the other side here because the loop is all crammed up inside that cell. But as you follow down through, I think it's through number 3 there, you can see at the end of that process, we now have 2 pieces of chromosome. All right, we have 2 chromosomes, each one in each cell. Notice, though, again, that Mother Nature being so efficient, that while we're finishing up copying of the chromosomes in number 3, the cell is already beginning to divide itself. The cell doesn't wait around. We're going to see that again in eukaryotic cells, too. So as the chromosomes are settling down and their copies are being made, and they're finding themselves in opposite ends of the cell so they can be divided, the actual physical cell is starting to divide, too. It's a very efficient process. And then eventually those cells pinch in half and we have 2. This is a very, very simple form of reproduction that we call binary fission. Binary is 2s. One make 2, then each one of those is going to make 2, right? That'll make 2 cells. That'll make 2 cells. And fission is to split. Here's fission right there. Boom, just like that. Now, the advantage of this type of reproduction is you can make a lot of offspring very quickly. I have the numbers in my Bio 2 class, and I don't call them off the top of my head, but I believe it was something like in one day, an E. coli can make something like 2 million offspring. Give me 1 second. Let me look that up because I think I'm exaggerating that number a little bit. But let me look it up, and it's a really interesting number because it says something about where they get their variability, because all organisms need to be variable, right? If they're not variable, then that means everybody's the same, and what kills one is going to kill everybody. So the question is, with many times with students, is, well, where do prokaryotes, since this is mitosis and exact copies, where do they get their variability? Where's that number? Where is that number? Hold me 2 seconds, right? I guess it's not there. But anyway, it's somewhere in the order of hundreds of thousands or maybe millions of cells. And what I want you to understand about this idea of making a copy of a chromosome, when you make a copy of a chromosome, you want to make an exact copy. You don't want the chromosome to be changed in any way. All right, and I say that with kind of a caveat, and what I mean by that is when you make copies of chromosomes, and we're going to talk about that in a few chapters of DNA replication, it's not perfect every time. We talked about back in chapter 3 where the adenines bind to the thymines and the guanines to the cytosines, and then that sequence of letters is your genetic code. Well, every time we make an exact copy of the DNA molecule, it does not turn out exact every time. It's just random probability. Every once in a while, there are mutations, changes in letters that are introduced into the chromosome. So, and this even happens in your cells when they undergo mitosis. Now, the benefit in our cells and in our chromosomes, there's a lot of our chromosomes, a lot of the information in our DNA is what we call silent. not that it doesn't speak or anything, but it's genetic information we have that that we don't use. I can explain this a little bit later when we get into the genetics chapters, but a good chunk of our DNA, an overwhelming proportion of it, is just sequence of letters that mean nothing. They don't code for any enzymes. They don't code for anything at all. If you're wondering why the heck are they even there, well, if you read Richard Dawkins' book called The Selfish Gene, it's a small book, but he talks about the fact that when chromosomes make copies of themselves, that molecule having the ability to do that, and that's kind of unusual among molecules to make copies of themselves. It makes copies to suit its own needs, right? It's just making a copy of itself. So when our chromosomes make copies, all the silent parts that we don't read, and then the smaller part that we do use, that is us, it's also read too, okay? But if in a bacterial cell or in a prokaryotic cell, they don't have the silent parts. So any mutation in the DNA has the effect, or potentially has the effect, on changing the cell. Now, most of the time, when you change the genetics of a cell, when you go in and you monkey with those letters, you kill it. That's right. Most of the time when DNA mutations occur, when letters are switched or during the replication process, that cell is not viable. It does not survive. Most of the time, an overwhelming proportion of the time, that cell, because the way it is with genetics is little tiny changes can sometimes have very profound effects on the way proteins are constructed and the way proteins work. And we all know that proteins are about shape. So most of the times those mutations are detrimental. They're bad, but not all the time. As I said, most of the time. I didn't say all the time. And if you reproduce a lot like E. coli does, which again is in the order of hundreds of thousands or millions a day, you can play the probability game. And if you say, well, 99% of any of the mutations that occur, and they're not going to occur that often, 99% of them are going to be bad. But if one of them just happens by probability to be a good mutation, like, for example, it makes E. coli resistant to some sort of antibiotic, then that mutation would be carried on in the future. So that's where they get their variability. They get their variability, these prokaryotes, simply because of the fact that the DNA replication process is not perfect each and every time. And whereas it's not perfect each and every time with us, it oftentimes happens to the parts of our DNA that aren't read. No big deal, no problem, no harm, no foul. Prokaryotic cells don't have that part of their DNA. Okay, their DNA is all red DNA. So if there's a mutation that occurs to their DNA, there's a very good chance that that's going to cause some sort of a genetic change to that cell. Usually bad, like I said, but if you reproduce a lot, you can play the numbers game. All right, that's all I want to say about prokaryotic reproduction. It's very simple. Binary fission, split the chromosome, make two copies of it, split the cell. All right. Any questions on that? Because I want to turn our attention now to eukaryotic cells, a little more complicated. How many chromosomes, speaking of eukaryotic cells, how many chromosomes do our cells have? We're a eukaryote. How many chromosomes do we have? I think I just mentioned it a second ago. A little bit more. 23 and 23 pairs of chromosomes. We really have a total of 46, okay? And Ryan is correct. We have 23 pairs of chromosomes because when conception occurred, egg and sperm, right? 23 chromosomes came from dad, 23 chromosomes came from mom. And those united together to make you, right? So we have 46 total chromosomes, or 23 pairs. Now, we'll stick with the 23 pair terminology a little bit more so in the next chapter, because that's where that becomes more important. But 46 chromosomes, you got to make copies of all of them. And remember, in mitosis, we're just making flat copies. They're clones of one another, essentially. So let's take a look at a pair of eukaryotic chromosomes. The chromosome is the red thing. And the reason why it looks a little different maybe is because this is what we call a condensed chromosome. Chromosomes, when we're getting ready to undergo mitosis, this whole thing, we're going to be moving them around. it's easier, it's more beneficial if you move them around when they're condensed. And I kind of use an analogy in class of if you're moving between apartments. If you're removing your dishes or your clothes, there's a couple of, well, there's two ways to do it, basically. One's much less efficient than the other. You could go to one apartment, pick up a dish, and then go deliver it to the new apartment, and then go back and get another dish, right? That would eventually get all your stuff moved over. Or you could condense those dishes into a box and move them as the box, and that's much more efficient. So if that analogy makes sense to you, then that explains why we're going to condense these chromosomes. Because remember, chromosomes are sequence long molecules of DNA, and when they're not condensed, if we looked in the nucleus of a cell, if we could even see them, they look like all kinds of little threads that are all wound up amongst each other. It's very difficult to figure out what's going on there. So when we get ready to move them, we're going to wind them up. And I'll show you how we wind them up here in just a second. But not only are they going to be wound up, but as we go through this process, we're going to make a copy of them. And that's what you see here. You see an original chromosome, and you see an exact copy. Those two red things are chromosomes, or copies of one another. All right, so in the process, as we move into mitosis, since we're going to be just like in the prokaryote, we're going to have to copy all 46 of those chromosomes. So let's get a little of this terminology out of the way here, because it can be a little bit confusing, and it's important because I'm going to use it a lot as we go through it. All right, what's the difference between a chromatid and a chromosome? Nothing. Chromosomes are chromatids. Chromatids are chromosomes. It's not a matter of difference in the molecule. It's a state of being. All right, this is what I mean by this. When the chromosomes are by themselves doing their chromosome thing and the cell is doing its cell thing, then they're a chromosome. During the process of mitosis, when the cell is going to make a copy, as I said, we condense the chromosomes, and prior to condensation, we make a copy of the chromosome. When the cell has a copy of the chromosome laid next to it, and actually held together, you'll see by these proteins, these cohesion proteins, we call these a pair of sister chromatids. All right, so what's the difference between a chromatid and a chromosome? Nothing. Chromosomes are when they're by themselves. Chromatid is when they're a pair. As a matter of fact, they're called a pair of sister chromatids. Now look down below, and you might say, wait a minute, what does this mean then? This is calling both of these things a chromosome. Think about a chromosome as being a unique set of information. Now, other than the little mutations that I talked about earlier, though, a chromosome is a unique set of information. So those two molecules there, those two pair of sister chromatids, represent one chromosome's worth of information, right? If you took your notes from class today and you made a copy of them, you basically are making you have two copies of the same thing. So if someone said, do you have the class notes? You do. You have two copies of the class notes, but it's still the same class notes, still the same information. All right, so chromatids, only when they're bound to their copy there, only when there are sisters bound together by cohesion proteins, and chromosomes when they're alone, or don't worry about what metaphase means. We'll get that in just a second. That's the stage that these chromosomes are in. Or one set of genetic information is a chromosome. All right, does that make sense? Because those chromatids, chromosomes, that can become very confusing. And I'll use those terms as we go through, and I hope to try to make them a little more familiar. Now, I'm going to come back to this slide in just a second, but I want to show you how this lumpiness all begins. In other words, here's a really cool picture. Again, chromosomes that they've made to fluoresce with different colors, and in this particular case, it's going to be more applicable in our next chapter. The blue ones are are what we call the paternal chromosomes, the one you got from your dad in the sperm. And the red ones are the maternal chromosomes, and the one you got from your mom in her egg. So you have, that's what I mean, 23 pairs of chromosomes. And there's a picture up above again showing you under the electron microscope these sister chromatids. The centromere, there is no real thing called a centromere. A centromere is a region. A centromere is a narrowing of the chromosomes, and sometimes it's in the middle, like you see here, and sometimes it's higher up on one end of the chromosome than it's the other. But let me jump to the next slide and then come back here and fill in the rest of these. We're going to talk here about how this comes about. How does DNA turn into this lumpy stuff? All right, so let's start at the top. There's our DNA double helix. Now, DNA is a very important molecule, very relatively fragile molecule, a molecule that you want to preserve. You want to preserve its structure. It's kind of like I mentioned, I think I did earlier on the semester, and if I didn't, it's kind of like if you were mailing china somewhere, glasses. You would put them in packing material. You wouldn't just throw them in a box. Just throw them in a box, they probably won't make it there. But you're going to put around all those, those peanuts, those styrofoam peanuts. So in the next level down below, it introduces you to something called a histone. Now, histone is a protein. All of this stuff inside of here is a histone protein. You notice the DNA is wrapped around it. All right, so we wrap the DNA around these protein histones as a way of protecting the molecule. Now, the DNA is not readable when it's like this. It can't be used by your body. But later on in a couple of chapters, we'll learn that when your body needs access to a particular set of genetic information, the DNA unwinds from the histone. That information is read, okay, and then it wraps around the histone again. So the histone is that white styrofoam stuff I was talking about. All right, now, groups of histones with a bunch of DNA wrapped around make something called nucleosomes, all right? And those nucleosomes in the third get coiled into this thing called a chromatin fiber. Now, then you jump down below, the little loops that are coming off of that red thing in the middle, this is called a scaffolding protein. Those loops that are moving around that scaffolding protein are made up of those coils of nucleosomes. And then a bunch of those loops together makes up that lumpy chromosome. So the picture you're looking at is a cross section. So that a red scaffolding protein would run right down the middle, and all those little loops would come off of it. And since those loops are so close together, you've got a lot of DNA, it looks like this big condensed thing. All right? Everybody see that? And of course, this is only when the chromosomes are condensing. This is when they're condensing, when they're getting ready to undergo cell division. All right, so now you know how those big red things occur. We talked about the cohesion proteins. Those are held together. We've talked about the centromere. Now, the centromere is the region. The structure at the centromere, this thing called the kinetochore, is a protein that is attached one to each sister chromatid. Right? You can see well, there's one there and there's one there. Those kinetochores are connected to things called kinetochore. Now, that's a new word, but this one is not. Microtubules. Remember in chapter 4, we talked about microtubules. Microtubules were part of the cytoskeleton. You remember the intermediate filaments and the actin filaments. And again, all those were components of the cytoskeleton, and they had all kinds of jobs. Amoebas used them for making pseudopods. Cells used them to form structure. And I think even back then, I know I mentioned that microtubules are also involved in chromosomal movement. All right, so this is what you're looking at here. There are going to be these kinetochore microtubules that are attached to the kinetochore, which are attached to each sister chromatid at the centromere, and those are held together by cohesion proteins. All right? So when you see these pictures in the future, you know what we're looking at. All right, so let's just follow one chromosome for a second, make it simple. Then we'll get a little more complex. We won't do it with animal or human cells. 46 chromosomes are just too difficult to put in any sort of a visual representation. It wouldn't look like just a whole bunch of chromosomes. But let's go with one. There are no organisms out there with one chromosome except for prokaryotes, but let's go with that. All right, so that chromosome, of course, is a double helix of the DNA molecule, and we're going to duplicate it. All right, see what's happened there? We made a copy of it. Now, there's a name for that in mitosis. When we've copied the chromosome, so when we've done this step here, actually I should say this step here, the cell has engaged in a process called karyokinesis. Karyokinesis. Now, kinesis, we know what that means. Kinesis is movement, right? Kinetic energy is energy of movement. Does anybody remember kary, K-A-R-Y, what that root meant? You heard it, prokaryote, eukaryote. No takers? Well, what do not quite, David. What do prokaryotes lack that eukaryotes have? A very distinctive cell organelle. It's one of the distinguishing features between them, right? What is that one organelle that prokaryotes don't have that eukaryotes do? And if you remember eukaryotes, EU means true, as in we really have one. Nobody? Nobody remembers that means nucleus, right? Eukaryote means nucleus. Eukaryotic, they do have a nucleus. Prokaryotic, no nucleus. Prokaryotic, pro means before, right? So there's no nucleus there. So this process of copying the chromosomes has a name that comes from that called karyokinesis, the movement of the nuclear material. In this case, the nuclear material is obviously the chromosomes that we're talking about. So, the movement is the fact that these chromosomes have made copies, and then as you see moving down into number 3, those chromosomes are going to separate, and then the cell's going to divide. And that has a phrase for it, and that's called cytokinesis. So karyokinesis has to happen first, right? In eukaryotic cells, you got to make a copy of the chromosome. Then cytokinesis happens next. Now, if you look at that, it makes perfect sense, right? It makes perfect sense to have karyokinesis before cytokinesis, because if you did it the other way around, you got one cell that's got the chromosomes and one cell that ain't got nothing. Without chromosomes, you can't survive, all right? So it's karyokinesis from step 1 to 2, and from step 2 to 3, the cell then divides. That's cytokinesis. All right, now cells aren't always dividing. Cells spend a lot of their time doing what cells do, making cellular products, regulating, doing all those things that we talked about in chapter 4. So this is what we call the cell cycle. So today we're going to discuss what's going to happen up here in the division, and we're going to discuss what happens in between as well. These stages that we call G1, S, and G2. We're going to talk about each of those. Collectively, those 3 phases, G1, S, and G2, are known as interphase. That's the first phase of mitotic division. There's going to be 5 phases, 5 more or less, 5.5, let's say, and interphase is the first one. Interphase is named because it's the interval when the cell is not dividing. You see, the cell is only dividing up here in the part that says the mitotic phase. During the rest of the time, in G1, for example, specifically, this cell is growing. See, the cell is just divided. So it's a smaller cell. You take one cell and divide it in half, it's small. Now that cell is going to grow, but only to a limit, right? It's only going to grow to the limit in which it remains efficient as far as moving things across the membrane. Remember, cells do stay small. Another thing that cells do during this G1 phase is they differentiate. In other words, what I mean by that, all the genes you have, minus the ones in your sex cells kind of things, but all the genes you have are in all the cells of your body. So in other words, the genes to be a liver cell are also found in cells of your heart. And the genes that make the heart a cardiac, or cardiac cells, are also found in the liver. It's which genes get turned on, get turned off. You see what I mean? So the whole library is there of genetic information. But if you're a liver cell, you're just pulling the liver book off of the shelf and following that. Or if you're a kidney cell, you're pulling the kidney book off the shelf and you're following those genetic information. That's what we mean by differentiation. During this time of cell growth, as the cells are dividing and getting bigger and bigger, each of the cells also begins to differentiate. Now, just because they're not dividing, though, doesn't mean that doesn't mean they don't realize they're going to have to divide again in the future. So as we move out of the G1 into the S, in the S phase, S stands for synthesis. During the S phase, the chromosomes start copying and condensing. All right, just like we talked about. So in other words, chromosomes are now going to start producing copies of themselves. They're going to start producing, and go back a couple slides, sister chromatids all over the place. In an animal, or in a human cell, this is going to happen among 46 of those chromosomes. Now let's see if you understand chromosomes and chromatids for a second. So let's look at a human cell. So let's imagine a human cell has finished replicating, making copies. It's finished condensing, and all 46 chromosomes look like what you see in this picture here. If we have 46 chromosomes at that point, how many chromatids do we have? "46 chromosomes. Each one has made a copy." Get it again, David. 46 chromosomes. Each has a copy. If we have 46 chromosomes, how many chromatids are there total? There you go. Right? So 92 chromatids from 40. Does that make sense, Ryan? 46 chromosomes, each with a copy, so we have a total of 92 chromatids. It's important, again, to be able to differentiate between chromatid and chromosome, because those words sound very much alike and they can be easily interchanged and you confuse yourself. So let's go back to S. So that's what's happening in S. The S phase, we're starting. It takes a while. It takes a while to make a copy. Again, you want to make an accurate copy, as accurate as possible. And there are even little things that go along the DNA when it's making copies. They're kind of like the old hand cars on the railroad. Proteins that move down the DNA try to find mistakes and fix them. Doesn't find them all the time, but you know. So that's all happening in S. Now, these are all dynamic phases. So is the cell still growing in S? Yeah, it might be growing a little bit. But this is really more applicable when we move into G2. So during G2, no, not really cell growth. During G2, what happens is the DNA synthesis is starting to finish up, right? We got them copy 46 chromosomes, so we're copying them. We're getting them stuck together with the cohesion proteins. We're getting them all arranged to make this copy, and then we move into G2. G2, it says there's cell growth, but I like to think of G2 as the stage in which the cell is getting ready to divide. And what I mean by that, it sounds very vague. The cell is going to start producing a lot of microtubules. You saw those earlier in a couple of pictures. It's going to start forming microtubules, and it's going to store up some excess energy, or not excess. It's going to store up some energy in the form of ATP to do this, because this is a process that requires energy. So all of that is going to happen in G2. So the cell might still be growing some, but the real significant thing about G2 is all getting ready to divide. So all collectively, that's known as interphase. And a cell in interphase on the left-hand side during G2, so this is right before it's getting ready to divide, is going to look like something you see on the left. The chromosomes are duplicated. They're in the process of condensing. So it says they're uncondensed right now, but they're going to. Remember, this is all happening dynamically. So during the S phase, they start to copy and they start to condense. In the G2 phase, they're still copying and condensing. They're going to be finished doing that copying and condensing at the end of G2, but it is going to still happen. Mother Nature is very efficient. Lots of things are going on at the same time. Now, let me point out a couple of things, the things that are in red. There are differences in the way this process occurs in animals and plants for reasons that I'll talk about a little bit later on. They're actually kind of interesting. But in animal cells, that's the part in the red. In animal cells, an area, not a thing, but an area called a centrosome makes a copy. So it makes a copy of that area. And that centrosome had a pair of centriole pairs in there. They look like little beer cans arranged 90 degrees to one another. So during this G2 of interphase, as we're getting ready to copy, in animal cells, and animal cells only, not in plant cells, the centrosomes copy, and that also copies at the same time the centriole pair. So now we have two pairs, okay? A little bit later on, you can actually see it here too. They didn't label it. This little, looks like a starburst. Those are only found in animal cells too. Now, the little starburst arrangement is called an aster, appropriately enough, because that means star, doesn't it? Aster, asteroid, astronomy. That's only in animal cells, not in plant cells, okay? All right, now, look what's going on as we move from G2 of interphase into prophase. So first of all, the chromatids are all condensed together. There's two sister chromatids of one chromosome, right? So this particular organism has, will you do it for me? How many chromosomes are being shown in this picture, in this model cell? Yeah, 6. How many chromatids? 12. Very good. Excellent. All right. So each sister chromatid is bound to its copy by the centromere. All right, now up above. In the animal cells, the centrosomes and the centriole pairs have replicated, and they begin to move to opposite sides as we're moving into the first phase. We're actually moving into mitosis now, moving into prophase. A lot of stuff is going on in prophase. The other stages are going to move pretty quickly. A lot of stuff is going on here in prophase. What does happen in plant cells and animal cells is those mitotic spindles. Now, let me give you some other terms for those. Those are also called spindle fibers. Anyway, those yellow things, those mitotic spindles or those spindle fibers, they begin to form between those two centrosomes in an animal cell. In a plant cell, they form, but there's no asters, there's no centrosomes. And again, I'll talk a little bit later about why animal cells have them and why we think plant cells do not. But this part right here, these early mitotic spindles, that also occurs in plant cells. The only part in animal cells is the part that's in the red, okay? So at the end of prophase, let me show you a picture. We got something that looks like this. Now, I'm sorry, this is not a plant cell. This is, or an animal cell. This is obviously a plant cell. You can see a cell wall around it. As a matter of fact, this is an onion cell. So at the end of prophase 1, we say that the chromosomes have condensed. When we look under the microscope here in lab in a couple of weeks, when we look at the onion cells in mitosis, you really can't see the chromosomes like you see in this picture until they're condensed. They're very thin threads, so when they're not condensed, they look like just a mass of maybe a mesh of these little thin threads of DNA. But when they begin to condense, they get bigger, they get thicker, and it's easier to see them. So you see here in prophase, we got the nucleus, chromosomes are condensing, right? And there's the nucleolus too. Nucleolus is not divided in this, but they're just identifying it so you know what it is. You see those chromosomes condensing? If you look to the cell just to the left, right there, you can see one that's not quite as far along in condensing, right? You're starting to see the lumps. I always tell my students when we look under the microscope, they look like little purple worms or spaghetti strands. They're purple because they were stained purple, not because they're normally purple. All right, so we're still in prophase. Now this is one of those half stages, right? Remember these, when I took genetics in college and advanced genetics, we learned something like 16 different stages. You guys were only going to have 5.5. But remember, these stages are dynamic. It's not like one ends, the next one begins. One is ending as the other one is beginning. Very, very, very dynamic, very efficient. So the next stage is kind of one of those intermediate stages known as prometaphase. Now, on your lab practical, when we do the cells and in the exam, I'm going to expect you to be able to look at cells and know the stages they're in. Don't worry about prometaphase. Prophase or metaphase, yes, but the stage in between, it's hard to tell under a microscope. So don't worry about that, but I do want to explain it to you. In prometaphase, you see all those sister chromatids are lined up and everybody's looking good. The nuclear envelope is going to begin to disappear. That membrane around the nucleus. We got to do karyokinesis. We got to move those chromosomes around. So we got to get that bilayer of phospholipids out of the way. So it'll come back, but we're going to get rid of it for now. And again, this is obviously an animal cell. You see the centrosomes and the pairs of centrioles. But now I want to talk about those microtubules. There's two types of them, you'll notice. Some of those microtubules are kinetochore microtubules, and if you notice, they're attached to the kinetochore. All right, this is the one we saw in the picture a few slides ago. Other microtubules are non-kinetochore microtubules, or I like to think of them, or not think of them, but I like the other term for them, polar. Polar microtubules don't connect to the chromosomes. Now, they have a purpose we'll get into in a minute, but the reason why I like to think of them as polar is the polar microtubules go from one pole of the cell to the other. Now, it's not one continuous. They basically start from, let's say, the south and the north pole, and they meet in the middle. See, they're kind of overlapped right there. But the important thing is they're not connected to a chromatid. They're not connected to a kinetochore, okay? And prometaphase, yeah, it looks something like that, right? Now, in lab, we'd call that prophase. It might be prometaphase. It very well could be prometaphase, but with our microscopes, it's going to be really hard for me to tell if the nuclear envelope has disappeared and whether all of those different types of kinetochore microtubules have formed. So we're just going to stick with calling this prophase. Now, in the next stage, as we move from prophase to metaphase, meta means middle. As I spell it incorrectly, meta means middle. So in the next stage, what happens is these pairs of sister chromatids, or this chromosome, they're going to migrate. They're going to move until they line up on the middle of the cell, this thing called a metaphase plate. Now, metaphase plate it's imaginary. It's sometimes it's called a cell equator. But the chromosomes, those 6 chromosomes, are going to line up along that metaphase plate. They're going to get in the middle. Each one of those is going to be connected, each chromatid, by through its kinetochore microtubule, in the case of an animal cell, down there where the centrosomes and down where those pair of centrioles are, whether the ones on the bottom and the ones on the top. Notice, though, that there are a few of those non-kinetochore or polar microtubules. There's one right there. There's one right there. So in metaphase, there's a mixture of kinetochore microtubules and those non-kinetochore, those ones that are called polar. Here's a cool picture of it, actually, again, with the fluorescence. Isn't that neat? These are what? Rat kangaroo chromosomes. The red, so the blue's the chromosomes, and the red are the spindle fibers, or the microtubules I was talking about. I just love this stuff, these great pictures. Here's another one, actually. Look up above. That quite clearly, if you look at it, for reasons that, as we go through lab, will help you understand, that clearly is an animal cell up above. And the reason why I know that's an animal cell is because it has these. Those are asters. I can't see the pair of centrioles, and the centrosome is just a region, right? So I can tell that this is an animal cell because it makes those extensive asters, those rays that come out from that centrosome region. Now, when we look through a microscope, it's a little bit misleading to think that the chromosomes are going to line up nice and even along a line like that, because you got to keep in mind the cell is a 3-dimensional object. It's not 2-dimensional. So those cells can line up in the middle, but not on a line, more on a plane, or something that we call the metaphase plate. So when you're looking down through a microscope, like in the picture up above, it doesn't look like they're all lined up in the middle, but they are. Some are below, some are off to the side, and some are up above, right? So some are at the top of the cell, some are at the bottom of the cell, and but they're all in that plane that is the middle of the cell, okay? And like I said, when you look through the microscope, you get, I think, a little better feel for what I mean by this, because they're not going to be all lined up perfectly. So let's zoom in a little bit on these lined up chromosomes. So again, in the upper right, there's the picture, and you can see once again, we have the metaphase plate, the sister chromatids, there are kinetochore microtubules, and then there are non-kinetochore microtubules, there are centrosomes and asters, so this is an animal cell. You can see on the left-hand side, once again, that the little picture on the bottom, that's an animal cell, because I can see asters. And it even labels the centrosome for you. All right, so we're all lined up in the middle here. Under a microscope, when we look at our onion cells, it'll look something like that. So if you're looking at that, you go, well, are they all in the middle? There's all these pieces hanging out here. Remember that these chromosomes are molecules, and they're all on that metaphase plate, but they all might. I tell my students, imagine if you're looking up above at a clothesline with a bunch of clothes on it, and all of the clothes are on the same line, but the wind is blowing the tails around. So they're all lined up on the clothesline, but the movement causes them to look like there's, pieces that are hanging out from the middle. But that's metaphase. Very easy stage to remember. All right, now we're ready for the dynamic part. Any questions on all this? So we've got chromosomes. We've made copies of them. We've moved them to the middle of the cell. We've constructed kinetochore microtubules. We've constructed non-kinetochore or polar microtubules. And of course, if you're animals, you have asters and centrosomes. The next stage, what's going to happen, I got to go back a little bit. Right. In the next stage, so imagine this is a pair of chromatids lined up on the metaphase plate. An enzyme is going to come in and digest that protein. Now, those kinetochore microtubules, I didn't tell you this before because I'm not sure it was in the right context, but those kinetochore microtubules, when they form, when all of this structure forms, they're under tension. They're pulling. They're already pulling. They're trying to pull these chromatids apart. Remember, it's cellular reproduction. It's making copies of the chromosome, so it's wanting to split those copies apart so each cell gets a copy of it. Those cohesion proteins, even though it's under tension, are what hold them together. In metaphase, an enzyme comes in and chops this up, chews it away. So now that the protein is gone, those kinetochore microtubules that are under tension, they start pulling. All right, and that stage is known as anaphase. Metaphase. Everybody lined up. Everybody ready to go? There's metaphase. There's anaphase. And so we pull them apart at anaphase, and now we're back to calling them chromosomes again. The sister and the daughter. Sister in this context refers to when they're attached to one another, and daughter refers to when the copies are no longer attached. So during anaphase, what we say is sister chromatids separate. Let's say that again. Sister chromatids separate in anaphase and become daughter chromosomes. Okay? And it is indeed those microtubules, those kinetochore microtubules you'll see attached to each one that are pulling that apart. See, they're the things that are actually moving the nucleus apart, or the nuclear material, the chromosomes apart. Now, what about those other microtubules, those polar ones, these ones here, the non-kinetochore? Well, what they're doing is, it's kind of like, no, this picture's better. Here it is. It's kind of like this. Up at the top is those polar microtubules are overlapped. All right? As the cell divides, the kinetochore microtubules are pulling the chromosomes apart. The polar microtubules are unoverlapping. They're beginning to push apart. You see down below? See how they're much more overlapped in metaphase, and we move into late anaphase, how they're much less overlapped? The overlap, again, is that dark area in between. What those polar microtubules are doing is, while the kinetochore microtubules are pulling the chromosomes apart, the polar microtubules are spreading the cell, pushing the cell apart. See, we're eventually going to want to chop the cell in two, so we're beginning to stretch out the cell, so the cell is going to take kind of an elongated shape to it. Anaphase is going to look like this under the microscope. You can see there's separation in between them. Can't see the polar microtubules, but that cell is being pushed apart. If we go back to an animal cell for a second, we get something that looks like this. Now, this is the next stage, but this is the result of those polar microtubules unoverlapping. We're pushing the cell apart. All right, so as we move from anaphase into telophase, anaphase ends when the chromosomes, the daughter chromosomes, have finished their migration, have finished their movement to opposite ends of the cell. So the kinetochore microtubules pulled the chromosomes apart. The polar microtubules push the cell apart, and the cell's elongating. And when we move into the next phase, got to have a nuclear envelope. As those chromosomes are being separated, and as they're being in each individual cells, they're already going to start uncondensing, decondensing, whatever the word is. They're already going to start unwinding. You can't read a condensed chromosome. The nuclear envelope is going to begin to form. The spindle fibers, the kinetochore, the microtubule kinetochores, and that we were talking about, they begin to disappear. Their job was just to move the chromosomes around, and that job is finished, so now we can get rid of them. Now, again, this is an animal cell because you see the centrioles and the asters. And eventually, if we look at a plant cell, we see something like this, right? This is telophase. We got two nuclei, but we have a cell. See, this one hasn't gone through G1 yet, has it? Yet notice these cells are about half the size of the cells in the previous picture, so they're going to grow after this. So in this telophase stage, again, it's two things that are happening kind of at the same time. The chromosomes are decondensing, the nuclear envelope is reforming, the spindle fibers are disintegrating, and then the other part of this is, as telophase is finishing up, cytokinesis is starting. You can see it right here. This is cytokinesis. This is an animal cell. And that works like the picture I showed you at the beginning of the class, right? You're basically, it's a bunch of proteins that act like a cinch, and they cinch around those two cells and pinch it in half until you become two cells. Okay, now back to the animal and the plants and the difference with the centrosomes and the centrioles and the pairs and all that sort of stuff. For a number of years, we weren't exactly sure why animal cells had these centrosomes and centriole pairs, and plant cells did not. But if you look at the picture of the way it's arranged, where's that picture I'm looking for? Yeah, right about there. It almost looks like you could assume that in animal cells anyway, that the centrosomes and the centriole pairs kind of act like an anchoring point for the kinetochore, right? I mean, if these things are pulling the chromatids apart, then it would be beneficial to have an anchoring point or a destination point where those kinetochore microtubules are going to be pulled to, right? And that's what that does in animal cells. The reason why we don't find them in plant cells goes back to chapter 4, because plant cells have a cell wall. They have a rigid cell wall. Animal cells only have a membrane. If you tried to attach these kinetochore microtubules to the membrane, you know, the centrosome is not there, the asters aren't there. As they pulled, you would pull in the membrane. The membrane is very flexible. In plant cells, what we believe, the reason why they don't need centrosomes and centrioles is because the microtubules, the kinetochore and the non-kinetochore, attach to the cell wall. So having the presence of that cell wall provides the rigidity for these things to pull. Animal cells don't have a cell wall, so we believe the centrosomes and the centriole pairs act as that. All right, so let's see, where are we here in telophase? Cells beginning to divide. That's an animal cell, and that's a plant cell. Now, they divide in two different ways, animal cells and plant cells do. You can see this is an animal cell. An animal cell is going to divide by a process we call cleavage furrowing. And it's what I just kind of explained to you. You basically take those proteins, and they're going to pinch and pinch and pinch this contractile ring of microfilaments or proteins, eventually going to pinch that in half into two daughter cells. That's cytokinesis. After karyokinesis, right? Got to have karyokinesis, then cytokinesis. Can't do it the other way around. Now, the way a plant cell does it is a little different. You see that in the picture. A plant cell forms, you can almost see, these are the microtubules that are disappearing. Let me erase that, though, because I want you to look at this right there. That faint line, it's called a cell plate. Because plant cells have a cell wall, they can't divide by cleavage furrowing. They can't divide, forward or backwards, yeah, forwards. They can't divide by just being pinched in half like that. Right, here's another picture. There we go. Animal cell. See, it's been pinched down, almost just about to separate in two. And up above is a drawing out of your book. Plant cells have to make a cell plate between them. Next picture is a little bit better. What I mean by cell plate is if we took two plant cells and we looked at them, what begins to form are little vesicles, opening, perforations. Imagine you took a piece of paper and you took a straight pin and you poked a bunch of holes in the middle of it. Just kept poking holes along that same line. Eventually, those holes would merge. So eventually, these vesicles merge and they form an open space. And then, of course, they line it with cellulose and they make two new cell walls. Or not two new cell walls. Well, they do, one for each cell. So they form that new cell wall between those two nuclei. The cell plate that you saw in the previous picture, so this is what it's going to look like under our microscope, and that is a direct indication that that cell is in telophase and cytokinesis. That cell plate is really a series of little perforation openings that, like I said, you know, they're real small, and then they begin to grow together, and then eventually they separate the cell completely in half. So we have cleavage furrowing in animal cells. We have cell plate formation in plant cells. Now we'll get some practice in this in lab, but let's look at some animal cells. These are animal cells, and see if we can't pick out some of the stages that they're in. And let's go simple first. What stage is that? the little dark purple part of the chromosomes, and then you can even see the spindle fiber. What stage is that one that I circle? There's another one in the same stage. That's the only two on the page that are in that stage. Where are the chromosomes? This is the cell. Where are the chromosomes? Oh, here's one. They're lined up in the middle, aren't they? So when chromosomes are lined up in the middle of the cell, what stage are we in? Okay, but what's the stage? Now it's just the chromosomes are going to split, but what's the stage? Is it interphase, prophase, metaphase, anaphase, or telophase? Which stage are all those in? All three of them are in the same stage. Yeah, they're all in metaphase. That's right. How about over here? What stage is that in? David? I don't think that's the only one that's in that stage. What stage is that? When I mean stage, interphase, prophase, metaphase, that's okay. Interphase, prophase, metaphase, anaphase, telophase. Which one of those stages is that in? It's the one right after from before, right? These all combined together are going to pull apart. They're going to look like that. So what stage is that right after metaphase? There you go. How about, yeah, that one will work. How about this right here? Or even this one. Actually, there's a lot of them. This is probably your best bet for telophase. See that? They're about ready to split in half, aren't they? Yeah, and the reason why you say, well, how do you know those aren't two cells side by side? Well, because I can still see the remnants of spindle fibers that are disappearing. So it doesn't all happen at once, right? So what are these that I'm circling? The ones where they're not lined up. Sometimes or identifying the stages of mitosis is eliminating the ones it's not. It's clearly not metaphase, because that's a metaphase right there. It's clearly not anaphase, because that's anaphase. It's clearly not telophase. It's the stage that occurs right after that one in the middle that I just circled. What stage is that one in the middle? Or what stage is any of these that I circled before? Let me get rid of all my scribbling so you know what circling I'm talking about. This one here. Oops. There we go. There. There. They're not lined up. They're not pulled apart. This here, I don't even see them, so they haven't even condensed in this stage yet. So what stage is that in the middle? Stage is interphase right there. What are these then? I can see the chromosomes. They're starting to condense. There you go. There you go. All right, and again, the one in the middle is interphase because you just can't see the chromosome. All right, now we're going to finish up like we usually do in a lot of chapters. We're going to talk about how this is all regulated. From the very beginning of the semester, I've hammered into you guys about the idea that metabolic processes and all that stuff are regulated, right? They don't go on all the time. So you're not making copies of cells all the time. And it's regulated, as you probably would imagine, chemically, right? Because that's all we are. We're a chemical, this really cool chemical reaction that's going on. So let me introduce you to a couple of proteins that regulate all of this. There are the kinases, and I'm in the upper left if you wonder where I'm getting those words. Kinases and the cyclins. And the type of cyclins are cyclins known as CDKs. They're cyclin-dependent kinases. All right, so they're dependent on each other. And when a cyclin All right, and it makes this CDK complex. That together makes something called a maturation promoting factor. That's a cyclin and a CDK complex. And that's what allows the cell to start moving out of G2 into the mitosis. So let's look over here for just a second. So here's the cell dividing, or here's the cell getting ready to divide. There's synthesis, S. Here's G2, the cell's getting ready to divide. As the cell enters into G2, this chemical, this protein, cyclin, begins to accumulate. There's cyclin right there. And the other protein, this CDK, that cyclin-dependent kinase, it's there all the time. Cyclin accumulates as we move into G2. When enough cyclin accumulates, it binds to the CDK and makes that maturation promoting factor. That combination, this right here, stimulates the cell to divide. After the cell divides, the cyclin disintegrates or degrades, and the CDK is left alone. And so the cell doesn't divide. So if you look at this picture on the left, in the purple is the cyclin. As the cyclin concentration increases, the maturation promoting factor increases, the cell divides. Boom, everything drops. Cell does its thing for a while in interface, the cyclin increases again, that binds with the CDK, making the maturation promoting factor, that increases, after it divides, everything drops again. So it's in a cycle, all controlled by the accumulation of cyclin. As cyclin accumulates, the cell divides. After the cell divides, it degrades. Then it starts to accumulate again, it divides. So it's on this regular basis back and forth. And a lot of this is controlled by the organism itself, right? Some of this is controlled by releasing this cyclin into the environment so these cells can divide, because sometimes you don't want cells dividing. As a matter of fact, this whole cell cycle has got some checkpoints through it, things that look and make sure that this cell division is going to be efficient. All right, so let's take a look at these checkpoints. Now, what I'm showing you here is a cell that's gone through all the checkpoints. The last checkpoint that we will concern ourselves about is going to be the G2 before we divide. But let's look at these other checkpoints. All right, there's a G1 checkpoint. We're in G1, right? The cell's divided. In the G1 checkpoint, the organism is going to check for the nutritional level, the presence of growth factors. Growth factors are chemicals that basically relay a message to the cell that says, go ahead and divide, go ahead and make more cells. And the reason why nutrition is tied to that is if your body is having a hard enough time supplying food to the cells it's got, then you really aren't going to make any more cells, right? Can't feed the ones you got, you don't want to make any more. Now, cell size is important as well. If you don't have the right cell size, then it's not going to move on to the next phase as well. Sometimes cells actually enter into a G0 phase. That doesn't mean they're dead or anything. That means that they're going to do cell stuff for a while. They're not going to go right into S right away. They're going to stay in this G0 and do their cell work that they need to do. And then as it's time to replace them, then they'll open up and move out of the G1 phase into the S. Okay? All right, move into the S. S phase occurs. Move into the G2. There's the G2 phase that I was just talking about. This is when the cyclin is accumulating. At the G2 checkpoint, the cell is going to make sure that replication is complete. All 46 chromosomes have been copied. Not only have they been copied, they've been copied accurately. That's what the DNA integrity. Now, do mistakes occur? Do mistakes get through? Well, obviously they do. Do changes and mutations occur? Do they get through? Obviously they do, because we talked about that earlier. But for the most part, those things are going to be caught at the G2 checkpoint. And then there's one last one. It's called the M checkpoint. That's actually a very important one. The M checkpoint, let me go back a little bit, is going to make sure that every chromosome has a kinetochore microtubule attached to it. The M checkpoint is going to make sure that, well, this will work, is going to make sure that every sister chromatid that becomes a daughter chromosome has a kinetochore microtubule attached to it. What would happen, and this does happen sometimes, these checkpoints aren't perfect. What would happen if that sister chromosome or daughter chromosome, what if that kinetochore microtubule hadn't formed or hadn't formed properly? What do you think would happen to the number of chromosomes in each resultant cell? So that last chromosome on the right has nothing pulling it up in this picture. Now remember, its copy on the other side has a microtubule that's pulling it, and they're attached by a cohesion protein, but nothing is pulling that chromosome up to the top. So where are those chromosomes going to go? The bottom cell is going to get both of them, and the upper cell is going to be missing one. Now, if you Down syndrome, what is Down syndrome? What is the genetic basis of Down syndrome? Down syndrome is sometimes called, other way around. Actually, you have an extra chromosome. It's sometimes called trisomy 21. You have an extra number 21 chromosome. The way Down syndrome happens is because of this. Down syndrome occurs because a kinetochore microtubule didn't form properly and did not pull the chromosome to the other side, the daughter chromosome. So as I said, this cell down here ends up with an extra one of those, and this cell up here ends up with missing one. Yes, and Ryan, you're right. You can be missing a chromosome. You can have an extra one, but trisomy 3, chromosome 3, number 21 chromosomes. That's Down syndrome. So that's an important checkpoint, isn't it? You want that end checkpoint to make sure that everybody gets the right number of chromosomes. All right. What happens when cells don't listen to these growth factors? What happens when we have something called uncontrolled cell growth? Well, we know that is another name. That's cancer. You see, cells have a couple of characteristics. One of them is what we call anchorage dependence. Cells require a surface for division. They have to be on something to grow, all right, some sort of a substrate. Many times in animal cells, that's going to be like a collagen foundation. And they're going to grow until they form a single layer. So they're going to grow in a single layer, and that's because they have density-dependent inhibition. Now, that's just a fancy word that means that the cells aren't going to grow on top of one another. They're going to grow in a single layer, and then if you have another layer of cells, they'll grow in a single layer. Then another layer of cells, they'll grow in a single layer. When cells become cancers, and there's a genetic component to this in many cases, many times it's a genetic component plus an environmental component, cells lose that inhibition, that density-dependent, and they become cancerous cells. They grow in lumps. See, that density-dependent inhibition works very nicely because if there's a gap in the cells, right, if there's an injury that occurs, the cells then divide and they fill in that gap. Still in a single layer, like you see in the picture. Cancerous cells are transformed genetically, so they no longer have that density-dependent inhibition, and they begin to grow on top of one another, and they form tumors. That's where tumor is a mass of cells that are growing in sort of an unorganized group. Now, the problem with tumors is tumors are cells that need energy, and they're going to start drawing that energy from the surrounding healthy tissue. So, for example, if you had like a breast tumor, like many females and males, 1 in 1,000, it's 12.5% of U.S. women will come down with breast cancer. That tumor begins to grow that mass of cells, and it begins to destroy the surrounding tissue, causing it to die because it's removing the nutrients from it. The surrounding tissue becomes necrotic. There we go. Necrotic. Becomes dead, dying, and that tumor begins to grow and then invades all that healthy tissue. And then if that tumor happens to be a malignant tumor, there's tumors that are benign. Benign tumors are cancerous tumors that stay where they are. Malignant tumors are the difficult part. Malignant tumors are the ones that folks who have gone through cancer treatment have to worry most about. Malignant tumors are tumors that can undergo something called metastasis. And metastasis means that cancerous cells can actually spread through the blood vessels or even the lymphatic system, move throughout the body, and then set up a cancerous tumor in another place. So that's the problem with malignant tumors. And that's why when they do a biopsy on a tumor, it's important to find out whether it's benign or malignant. If it's benign, then removing it pretty much cures you from that particular cancer in that spot. Malignant, if a tumor is malignant, you can remove that tumor, but you don't know necessarily at the time if it's spread. That's why a lot of cancer patients will talk about 5 years the rule of thumb is if cancer doesn't show up again in about 5 years, then you can assume that your malignant tumor, now they'll know that tumor is malignant when they pull it out and biopsy it. You can assume that that malignant tumor hadn't spread yet, hadn't begun to engage in metastasis. That's a rough rule of thumb. My mother had breast cancer and 10 years later died of bone cancer. Her tumor was metastatic. She had gone through the 5 years and figured she was clean, and then about 5 years later, she came down with bone cancer, and bone cancer eventually killed her. And the bone cancer was a result of the malignancy of the breast tumor that she'd had 10 years prior. So it's no guarantee to 5 years. See, that's the problem with cancer. The benign tumors affect the local area, and you can oftentimes remove them, and many times it's not a big issue. And that's many times when women have mastectomies, have their breasts removed because of breast cancer. What they'll do is they'll remove all the breast, the tissue, and part of the lymphatic system. The idea behind that is if it didn't metastasize, you hope that you get all of it. So where it spreads through in the lymphatic system, you remove a lot of those lymph nodes, maybe you got those metastatic cells before they can spread around the rest of the body. Any questions on any of that? All right, so that's it for chapter 10. Let me look at something. Next week we have to do our lab practical. Let me look at the schedule a second. I think I'm going to announce this now while it's on the recording. Lab practical next week, chapter 11. Do that up. You know what? And I'll put this in an announcement. We're not going to have lecture Monday morning. We won't have lecture Monday morning. What we'll do is we'll just start the lab prac at 9:30. Let me write myself a note. So does everybody understand what I mean? So no lecture Monday morning. I will be in the lab if you have questions, but we're not going to hold lecture. We're just going to start. Give you a few more minutes to, well, another hour and a half to study for the lab practical. But then we'll start the lab practical promptly at 9:30. Any questions? What is it, 1 through 4? Yeah, take a look at, or 1 through 5. Take a look at what we went through in the lab. Look at those questions that we answered in the lab. Make sure you can do those problems like the C1, V1, all those ones that you gave practice problems to. If you concentrate on the information in the lab, now don't worry about running that Logger Pro software. I'm not going to ask you information about, how do you set the experimental time or anything like that. But I might print out some graphs from there and ask you to interpret them. Like I might say something like this, and let's say this is temperature. And maybe this is enzyme reaction rate. And it looks like this. And I might ask, why does the reaction rate go up? What is the optimum? Why does the reaction rate go down? Right? For example, something like that. You see what I mean? So I'm not going to ask you to generate graphs. The only thing that you might have to perform to do is I may put in a question where I ask you to build a functional group. So you rotate to a station, and I'll say, okay, build me an aldehyde. Or I might have a station where there's a microscope, and I ask you to get that paramecium or whatever's under the microscope and focus at 40x, and you have to go through all the process that we learned in lab 4. But and as I said, focus on those questions that are in the lab, throughout the lab and at the end of the lab. You do need to know some formulas. You will have to memorize those. So density, C1, V1. Oh, and the one for magnification, M1 times DF. This is in lab 4. M2 times DFV. That's the other one. All right. Okay, folks, if you don't have any other questions, that's it. I'll see you guys Monday at 9:30 then.